But I want to test everybody. Does everybody remember Kowalski's rule of three? A little test here. It's going to be a test. I don't know what it means, but linear standard curve. Number two, precise points of intercept. And number three, standard and samples have the same shape. That good enough for some catches? All right, thank you. Good. So we're going to have, I think that in terms of one main benefit of KinetiSol is obviously the solvent use compared to spray drying. And just wrote a couple of notes here that by 2026, it's expected the world's going to use about 33 million metric tons of solvent. The United States will take up about 4 million metric tons of that, and China, 12 million metric tons. That's growing at about a rate between now and 2026 of 3.4%. The dollars are out there at about 8 trillion or $8 billion dollars, but it's growing at a faster rate. It's growing at a rate of 4.8%. Compared to what I'd said was the regular solvent use in the world is 3.4%. You can say, okay, well, that's because we're all getting older, and it's becoming, pharmaceuticals and healthcare is becoming a bigger part of our world. It's true, but I wonder also if it's because we're using more solvents in the pharmaceutical industry. And 80 to 90% of pharmaceutical waste is solvents, apparently, and of course not just for spray dry dispersion. I hope it's not that, but the COVID, all the vaccines use them a lot. So I think this next speaker is going to be important to learn how we can have the possibility using KinetiSol in disrupting spray drying. We'll learn later on in the afternoon how it's not a one solution for everything. Absolutely. So we have to understand when we use it and when we don't. But in general, we'll be using some real world experiences and examples where KinetiSol successfully replicated commercial SDDs, offering benefits like the elimination of solvents and better drug properties. Comparative analysis will be provided, contrasting the infrastructure and processing needs with KinetiSol, offering insights into the efficiency and simplicity of the latter. And Dave will delve into the superior biopharmaceutical performance of KinetiSol over spray drying using multiple case studies. And as we learned from this morning, robust analytical data. And hopefully by the end of this next speaker, we'll be able to recognize the possibility of KinetiSol as a commercially viable method to simplify the manufacturing process. Yesterday, we were talking a lot about how it makes our job easier. And I think that's interesting because I always think, well, a new technology would make something harder. We know spray drying, this is different. But I'm interested in, for myself, how it can make our world our work a little easier. So I want to join in welcoming Dave Miller, the Chief Scientific Officer at AustinPx. He has a very rich background that includes a decade as VP of R&D at Dispersol and a significant tenure as senior scientist at Hoffmann-La Roche. He's contributed to over 42 research articles and 8 book chapters. And he's a proud alumnus of the University of Texas at Austin here, where he got his BS in chemical engineering and a PhD in pharmaceutics. So let's give a warm welcome to Dr. Miller.
Okay, so there's my title. It's pretty bold. Last week it was even more bold. We were on a virtual meeting with the other speakers, and it said, why KinetiSol will displace spray drying. And my good friend Michael Crowley audibly chuckled, and I said, well, maybe that's a little too bold. Maybe I should back off that a little bit. And because it probably, there's always a place for various drug delivery technologies. So I replaced displace with disrupt. And I think that's appropriate. I think that's an appropriate title because I'm not formally business trained, but I read a book from the Harvard Business Press once called The Innovator's Dilemma. And there's a lot of important lessons in that textbook about commercializing a disruptive technology, particularly in a conservative industry. And I think a lot of those lessons really read onto our history of KinetiSol. And I think the key is that my take-home message from that book with respect to KinetiSol was that you've got to stay flexible and adaptable, and you've got to withhold resources because your first thoughts about where your place will be in the market may not be correct. So you got to be able to respond to market feedback. And I think what we've done recently in pivoting to Austin Pharmaceutics and bringing this great business team and Tim in, we've eliminated a lot of the barriers that precluded some companies from utilizing KinetiSol. And in doing so, we've brought on more molecules in the last 6 months than we've seen in quite some time, and that allows us to get a lot of feedback from industry. And what the industry is saying, per analysis of our current pipeline, is that they're struggling with a lot of limitations of spray drying. There are many and various problems with spray drying, and they're eagerly and actively looking for a better solution. And that's the reason why about 3/4 of what we're working on today is KinetiSol conversions from spray drying. So I think this is an appropriate title. The industry is looking for disruption. We've evolved this technology over the last 16 years to a state of maturity where I think we're ready to take on the challenge. We're ready to implement KinetiSol commercially, and so we're at the right place at the right time to capitalize on where the industry is with respect to use of solvents and other limitations of spray drying. So in these next several slides, I hope to convince you all that we're on the cusp of KinetiSol making a serious dent in the spray drying market. Later today, my esteemed colleague, Dr. Justin Keene, will get into more of the nuts and bolts in the process. And tomorrow at the Technology Center, Chris's Chocolate Factory, I hope you all are going there. It's going to be a great experience. You get to see these machines in operation and how they're made. Chris is going to go into great detail about the technology, how it works, its history, et cetera. So I'll just gloss over these slides, but I think everybody here, if you're here, you probably know the basics of KinetiSol, but I want to calibrate the audience just with some brief overview. So I'll go through these slides really quickly. I think everyone knows when I met the process and when I met Chris, it was a mature industrial scale plastics processing technology. And it went through a drug-induced mutation over the last 6 years to becoming a very elegant and robust pharmaceutical unit operation. And here's a schematic. Colin and Rob have heard me present this so many times. It induces nausea at this point, I'm sure, but you're going to appreciate this. So I'll leave the details to Chris, but essentially KinetiSol is an ultra high speed mixing process. It converts a blend of crystalline drug substance and excipients into an amorphous solid solution, and it does it in about 10 seconds at temperatures well below the melting point of the API. And that's it. That's the fastest I've ever given that overview, and it pains me to leave out all the details, but Chris will do it tomorrow, and he'll do it much better than I ever would. Again, we have 2 scales of equipment, research and manufacturing. You'll see these in operation tomorrow, and Chris will go into great detail. For us, the research machine in my group is the workhorse. This is where we can establish feasibility with any given molecule with single-digit gram quantities. And if you've got 50 to 100 grams, we can exhaustively map a formulation space to find the optimum formulation. And we're even using this system now in GMP as a way to accelerate drug product into a human PK study. So eliminating need for engineering batches and expediting tech transfer. And then next, it's just simple linear scale factor to the manufacturing machine where, this is a very versatile machine, can produce first in human supplies all the way to commercial from a single machine in a very small footprint. So that's all I'm going to say, and I'll leave the rest to Justin and Chris. So when KinetiSol came on the scene, eliminating the use of solvent and minimizing thermal stress through both time and reducing the duration of exposure, reducing temperature, it really shifted the boundaries for ASDs and allowed more incorporation of more actives. So, high melt point, or organic solvent solubility, thermally labile actives. Now, more of those have become viable in the ASD space. And similarly, it expanded the excipient space for a lot of the same reasons. We could process polymers that were thermally sensitive or non-thermoplastic or didn't have a common solvent with the API. So it really does broaden the formulation design space relative to other technologies, and that creates opportunity for more optimal drug products, surprising results that lead to meaningful IP, compositions that can't be duplicated by other technologies. So I think it's not trivial that the operation of the process and how it increases what you can do in terms of formulation for ASDs. It's not a trivial thing, and I'll show it later in the slides of how we've been able to use this broadened design space to really develop optimized ASD products that have real clinical benefit and bring along with it some significant IP. So when we first sort of came on the scene, this was a diagram that was presented by several different scientists around the industry, and in the top quadrant where these brick dust compounds were and starting to emerge back in the late 2000s, there was a question mark in this box that these brick dust compounds are becoming more and more prevalent. We don't have a really great solution. We need some innovation in this space. So this is kind of where we focused initially. Really thought that KinetiSol as an enablement platform was going to make a huge impact, and it still has that potential to really impact brick dust compounds. But what was surprising was the application of KinetiSol in the other quadrants, where melt extrusion and spray drying have been the more conventional approaches. There were some really key limitations in each of these quadrants where KinetiSol was a more viable option when solvent processing is an issue, or where thermal degradation limits melt extrusion, or viscosity limitations impact formulation design. So where we focused initially on enablement, I think we've seen a lot more application in the other quadrants, just as a result of there are more compounds in those spaces than there are brick dust molecules. When we look at our development history as a function of melting temperature, I think what stands out is that about 73% of the compounds we worked on historically would be considered high melt temperature. Melting temperature is greater than 200. And maybe even more surprising, 40% have melt temperatures greater than 250. And I think we may all agree that melting temperature kind of loosely correlates to all solubility. So many of these compounds are also organic solvent challenged, they're solubility challenged. So now this data set's probably enriched given the technology that we have, but it may sort of represent also general trends in drug discovery that molecules are trending more towards high melt point, low organic solvent solubility. So starting to see gaps in the ASD coverage space with the conventional technologies, and a growing need for something like KinetiSol. And then now calling back to our Venn diagram here and the expanded formulation space of KinetiSol, and back to my point that this is not just a trivial thing that you can put more ingredients into the formulation. When we look at our clinical product history, the drug products that we put into the clinic using KinetiSol, 2/3 of those have been multi-component systems, and these components that we've incorporated into the formulation beyond just a drug polymer binary system have been key to first optimizing performance. So that's the goal: maximize bioavailability, and then other components are incorporated to ensure manufacturability and stability. And in a lot of these cases, these ancillary excipients have been key towards stability or performance to an extent that constitutes the surprising result of a formulation development, where you can get some meaningful patent coverage around an ASD that incorporates this drug, this polymer, and this stabilizing agent. And without that agent, you don't have a viable product. So that's where the IP component comes in, and also incorporating performance enhancers that allow you to achieve maximized PK at a given dose. So to the extent that it would be difficult or impossible for a competing technology to duplicate down the road, preventing an AB-rated generic. So with all that front matter out of the way, I'm going to descend into my title here about my contention that we're poised to disrupt spray drying, starting with a couple of examples of where we've shown KinetiSol to be a substitutable technology for spray drying for commercial products. And so case study one is KinetiSoling Intolerance. So Intolerance is a FDA approval in 2008. It's an HIV drug. It's a J&J molecule. What attracted us to this drug product as a company was that the patent exclusivity, the patents were up on the API in 2019, and all the regulatory exclusivities, pediatric exclusivities were expiring in 2022. So this was effectively one of the first spray dried products to come off patent. And we targeted it as a product that we could make an AB rated generic using KinetiSol. It was challenging to say the least. My longtime friend and colleague Sandra Kuchera courageously led this development. I fear it took years off of her life, but here's the reward, a really compelling case study for KinetiSol. So it's a spray-dried dispersion of etravirine in HPMC. It's a highly variable product, and bioequivalence is difficult to achieve even with the same technology. A little more detail, specifically, it's a 25% drug load SDD. The feed solution, the thing to point out here is it's a relatively low solids load. You know, you're spray drying, per this patent example, 600 kilos of solvent. A majority of that is chlorinated solvent to obtain less than 9 kilos equivalent of etravirine. So that's a problem, I think, in terms of, you know, the way we see green chemistry now and the ecological footprint. Like most spray dried products, it requires secondary drying and roller compaction. I've already mentioned that the drug product has highly variable performance, and obviously it's environmentally and economically very costly. So when we set out to work on etravirine, we were slapped in the face with some real challenges. It's a high melting temperature compound, 272. That's not unusual for us. As I showed in the diagram, we're used to working on a lot of high melting compounds, but the molecule degrades at melt. It degrades before melt, actually, I think. So the challenge would be, can we render this thing amorphous by KinetiSol and quench it fast enough before there was any degradation? It's a BCS4 molecule, so that presented some challenges as well in terms of trying to match insolence from a bioequivalence perspective. It's not just a matter of dissolution rates and solubility. It's about presenting it at the right place in the GI tract. And we're working with this high melt compound, we're working with a minimally thermoplastic polymer in HPMC. So not a lot of, I mean, it's not helping us out in terms of viscosity and polymer dissolution. And, you know, beyond all this, duplicating an amorphous solid dispersion made by a solvent process with a thermal process. It's like very dissimilar technologies trying to establish a bioequivalent product. It's a big challenge, but we were able to do it. Here's an example of the processing profile, pretty typical KinetiSol profile, as I mentioned before. In less than 10 seconds and at a temperature about 100 degrees below the melting temperature of etravirine, we were able to generate a single phase amorphous dispersion, matching the composition of the spray-dried dispersion. But I've laid out all the challenges from a biopharmaceutical perspective in trying to match the clinical performance of Intolence. And Dave, I'm gonna pad your stats here and give Dave K a lot of credit for the microflux. He came down, he trained, my whole team has been trained by Dave on the use of the flux system, and flux was really the critical tool. It was what provided the in vitro and vivo correlation that allowed us to be successful in the fasted state. So we leveraged that tool along with dog modeling and GastroPlus, and went through various iterations and ultimately arrived at this slide, which is the punchline. So we were able to develop the product. In our pilot studies, we were able to show bioequivalence to Intolence despite the high variability of the comparator product. We made an IR tablet through direct compression. As I'll talk about later, KinetiSol eliminates the need for roller compaction, secondary drying, et cetera. It's very straightforward direct compression. And we were able to scale this up to registration scale, as represented by this diagram here, which was our PAT tool. Justin will talk about this more in his presentation, so I'll just say this is the pictorial representation of registration scale KinetiSol, and Justin's going to talk more about the details. So that was a big success. We were able to show that KinetiSol is a viable substitute for commercial spray drying for etravirine. Now on to case study 2, and this is the subject of some fairly recent research, so we blinded it for now. We'll make it public soon. But the commercial product contains the API in HPMCAS, once again in a 1:3 ratio. Similarly, the feed solution to spray drying contains quite a bit of chlorinated solvent and ethanol and a fairly low solids load. So it's very costly in terms of spray drying in a lot of drying time, a lot of solvent waste to obtain relatively little amounts of API equivalent. And once again, a much more complicated downstream processing approach to achieve a tablet. So we were able to take this molecule, duplicate actually create a range of amorphous drug loads. So it works very well with KinetiSol. We were able to establish a range between 20 and 40% of single phase amorphous solid dispersions with absolutely no process degradation. In fact, we purified the API a little bit here as our process related impurities are negative. And then in terms of looking at optimization of drug load, we DISO screen, two-stage DISO, looking at our various drug loads relative to the spray-dried dispersion, and identified that at 25% we were slightly better than the spray-dried equivalent, but that appears to be the optimum drug load for this product. This is the one where Danny was saying he didn't want to use the TIM2, but I think we need to understand speciation a bit more here, and we'll talk to you, Dave, about that. So we took this optimized ASD intermediate and took it through our standard tablet manufacturing process, where out of the compounder, out of the compounder comes our larger cookie. So you've got all on your tables there, example of the discharge from the formulator. So this is what they look the same on the manufacturing scale with a larger diameter. And you take those through a milling process, and most of the time it doesn't make blue granules, but in this case they were blue. Then you blend them with external excipients and run them through your tablet press to arrive at your tablets. I think what's important to point out here is that we were able to arrive at tablets with acceptable tensile strength and disintegration dissolution performance with 85% of the amorphous dispersion in the final tablet, which will become important later in the presentation. But we're able to, this is key for this product because at the high dose, there's a pretty significant pill burden. The high dose product is very large and difficult to swallow. And for this particular patient population, dysphagia is an issue. So whatever we can do to reduce the pill burden is important. And by maximizing ASD load in the tablet, we're able to significantly reduce tablet size. So once again, using our Pion tools, we're able to show with pH change DISO that by developing a tablet, we didn't alter the dissolution profile of the ASD intermediate, and then running flux between the KinetiSol intermediate and the spray-dried dispersion, we showed very similar flux profile from our tablet. And then finally, this all culminated in an animal PK study. In this case, we did a rodent PK where we took the commercial tablet and pulverized it into a powder and dosed that against the equivalent of our KinetiSol tablet. And what you see sort of qualitatively, well, if you could see it, the PK profiles lay virtually on top of each other. And since that doesn't look so clear, you can look at this F%. We're about 96% of the commercial tablet. So there are some next steps involved here in further establishing this, but I think we're off to a good start in terms of establishing there's a high probability of success that we will be able to duplicate this commercial spray dried product with KinetiSol. So what I've shown here is 2 case studies where we show KinetiSol to be substitutable for commercial spray drying, and the implications are we would eliminate tons of chlorinated solvent and waste solvent annually. The technology obviously has a much smaller physical footprint. You've seen that our manufacturing compounders that you'll see tomorrow have outputs that rival PSD4s, maybe even larger, in a system that's on wheels and could be rolled into any existing GMP suite. You can operate multiple in parallel, so you don't have to build an entire plant around a KinetiSol manufacturing line. It works in existing GMP facilities, and obviously the ecological footprint, a lot less energy usage, no solvent usage. And also in terms of capacity constraints, I think that's one of the biggest problems that we see from the clients that approach us. They're in phase 1 or even later with spray dried dispersion, looking ahead and trying to figure out where they're going to manufacture their commercial product given the capacity constraints for spray drying. So that doesn't exist with KinetiSol. We make the machines, happy to make them for you, put them wherever you want, and you don't have to, there's no major CapEx risk. If you wanted to build your own spray drying plant, you're in hundreds of millions of dollars until your phase 3 fails, like with as was the Teleprovir story. That risk doesn't exist with KinetiSol. So, and I think by now I've shown it's a simpler, more efficient process. And with some of the benefits of the form in terms of maximizing drug load in the formulation and maximizing ASD load in the tablet. There's significant potential to reduce pill burden. So now I'm just been talking about making equivalent products to spray drying, where the goal is to be bioequivalent. But now I want to talk about the case studies that we've seen in collaboration with our big pharma partners, where we've shown superior biopharmaceutical performance and understanding why particle properties are critical. So early on in our history, we had a fairly extensive collaboration with Boehringer Ingelheim in Connecticut. And they originally came to us with the issue illustrated on the left. They were using SDDs in preclinical studies, and they were having issues with respect to suspendability and gavageability of spray-dried dispersions, batch-to-batch variability. The dose in their tox studies was being limited by what could be administered from suspension with an SDD. And as they got into chronic tox studies and the demands for drug product approach the 100 kilo scale, they were having issues with supply chain and budget. So they came to us with some very practical needs to ideally provide an intermediate that was more suspendable and gavageable, and to be able to manufacture at large scale to meet their timelines and their budget. And so the panel on the right is there to illustrate that we were able to tick all of those boxes. The KinetiSol intermediate typically has a bulk density between 0.5 and 0.6, which tends to be much better in terms of suspension and gavage dosing. We were able to manufacture the material a few batches for them at a fairly large scale and deliver it ahead of schedule. And I'm guessing it with a little lower cost than what they were being quoted from spray drying companies. But the surprise result of all of this, we were just asked to solve some practical problems. The surprise was when they took our KinetiSol formulation, which was quantitatively identical to the spray dry dispersion. When they dosed it at the maximum dose that they were able to administer from spray drying, they got a 4-fold increase in exposure. Obviously, there was some error in dosing or in the study, so they dosed it 2 more times and got the same result. So in triplicate, they got a result that demonstrated that the same formulation produced by KinetiSol provided 4-fold increase in exposure over spray drying. And more than that, they were able to escalate the dose well above the maximum dose they were able to administer from spray drying. And as this was presented in 2017 by Keith Horspool, who was VP at Boehringer Ingelheim at the time, and he presented several case studies. This was one of the first ones, but he said this was important to the company because this allowed them to identify tox issues that wouldn't have been otherwise identified, and they killed the drug. And he estimated saved the company about $10 million. Avoiding clinical failure. So similarly, we just did we came off a study recently led by our own Urvi Gala, where with Eli Lilly, they were facing similar issues. They were in preclinical studies. They were headed into a chronic tox study for a phase 2 drug, and the preclinical safety team they were faced with a problem that they weren't getting the exposures high enough. They weren't meeting the safety threshold with the spray-dried dispersion, and the formulators were shrugging their shoulders about, what more do you want us to do? We've got a spray-dried dispersion. There's not much more we can do here. Fortunately, Rob and I swooped into Indianapolis. We flew in with our capes on and showed them that Boehringer Ingelheim graph, and they said, if that's real, we really need you to help us out here. So we did, and we did. So we did the same thing, basically, effectively duplicating the spray-dried dispersion formulation. They went into their tox study, and unfortunately, they didn't see a 4-fold increase. They saw a 6-fold increase with the KinetiSol dispersion. And, obviously, very satisfied with that result. And once again, there was some concern, though, that our drug loading was slightly lower than the SDD, and we had a different vehicle. So they're in the process of repeating this PK study. So I'm really looking forward to seeing the 6-fold increase twice, much like Boehringer Ingelheim dosed it in triplicate. I think we'll see the same result again. But anyway, so we're starting to see a trend here. Here's another case study that Keith presented back in 2017. This time we've got some in vitro data to accompany the PK data. And what we notice here when comparing the KinetiSol process material, that's this I don't know if you can see it, this green line, versus the spray dry dispersion. So both versions of the spray dry dispersion show, even though it's an enteric polymer, it's all HPMCAS based, right? It should be protected from acid release. There's significant release of the drug in the acid phase, whereas with KinetiSol, you see almost no release in acid. And then as you transition into the neutral phase, this version of the spray dry dispersion begins to precipitate almost immediately, while this one shows some spring and then followed by fairly rapid precipitation. The KinetiSol formulation springs into solution. There is obviously a parachute effect, doesn't quite hit that spring and plateau that Dana was talking about earlier, but begins to precipitate. So you start to see the in vitro differences that start to lead you down the path of a hypothesis as to why KinetiSol is routinely outperforming spray dry dispersions. So sure enough, this translated into a canine PK study where once again we saw with different particle size versions of the KinetiSol material significant increases in exposure relative to the spray dry dispersion. Dana mentioned that there are some instances where the recrystallized spray dry dispersion performs worse than the physical mixture, and this is one of those. The dispersion actually precipitated out from suspension and in the gastric environment so catastrophically that I think the particle size, either the particle size increase or you've got aggregates that are even more slow to dissolve than the dispersed physical mixture. So we started to form a hypothesis at this point that it's got to be something to do with the surface area or the density of the particles, that it's this protection in the acid phase that protects the payload, the amorphous payload, for release in the intestinal tract, whereas with spray drying, the higher surface area particles, the drug's able to diffuse out of the enteric matrix before the polymer is soluble. So you've released your spring without deploying your parachute. But in the case of KinetiSol, your parachute is preserved until the spring releases in the neutral phase, and then you deploy them simultaneously. So to use the verbiage from the Taylor group these days, KinetiSol particles promote congruent drug polymer release, whereas in these cases, the spray dried particles seem to be promoting incongruent drug polymer release. So we, in collaboration with our good friends at the University of Texas and some buddies of ours at Merck, did a study to really sort of examine this effect in depth using BI's 639-667 compound, the same compound in this study, and just some basic properties of this molecule just to show it's a real compound. It was in development at BI at one time. So the methodology for this study was to produce the same formulation by KinetiSol and spray drying, and then do in-depth characterization at the molecular level and at the particulate level, and then look at performance both in vitro and in vivo. So first, looking at the molecular level, obviously first stop X-ray, they're both fully amorphous per XRD. Looking at FTIR, we saw no differences between drug polymer interaction. And the young Chow at Merck was very kind and did some very in-depth solid state NMR analysis that I'll be honest, I don't understand. So don't ask me a lot of questions. I can just say that he says between 1D and 2D solid state NMR, the samples are molecularly similar. And then when looking at miscibility, I know enough to say T1 and T1 row, those parameters are similar, which means that in both cases, the drug and polymer are completely miscible, in this case to a domain size of about 32 nanometers. So based on the solid state NMR analysis, XRD, FTIR, at the molecular level, there appears to be no difference between spray drying and KinetiSol. So there's no, at least per these analytical techniques, there doesn't appear to be a difference in how the drug is distributed in the matrix. But when we start to look at the particulate properties, you start to see divergence. With spray drying, you see much finer particle size, as you might expect, and a very high specific surface area that's about an order of magnitude greater than the KinetiSol particles. Even when we go to the extreme of cryomilling the KinetiSol material to reduce the particle size to a minimum, we're still only about half the specific surface area of spray drying. And we also wanted to examine the effects of how do you tune particle size for spray drying? Well, it's through compression. So we sort of simulated high and low pressure compression, and you see the specific surface area doesn't really change very much. And likewise, when KinetiSol particles can be tuned through the milling process and optimizing your milling parameters to achieve different particle size ranges. And within the typical range of particle size, we don't see the specific surface area change too much. So it's just inherent to the particles themselves. KinetiSol inherently has a low specific surface area. Spray drying has a very high specific surface area. I don't think this is, this all fits with intuition. We added a little bit of another layer to the story recently. We just published a paper with our collaboration with UT, looking at galeterone, and Stephen Thompson and did this research, and he looked at surface energy analysis by inverse gas chromatography for KinetiSol dispersions in HPMCS-H with galeterone versus the spray dried equivalent. And what was interesting about this study was that, as you can see between the left and right panels, the surface energy of the KinetiSol particles, particularly acid-base surface energy, is substantially lower than the spray-dried particles. And I think this is really interesting and something that we're just starting to think about. As you form a particle from a solvent-based system, there are interactions with the polymer and the solvent. As the solvent is being evaporated and the particle is starting to form and evolve, you can expect that perhaps there is some change, some specific orientation of the polymer, where polar groups of the polymer might find their way towards the outside of the particle, whereas with a KinetiSol particle formation, where it's sort of just a random mix of drug and polymer, those polar groups don't orient in a specific way. So that may explain this lower surface energy. It could just be a function of surface area as well. I don't know. Nonetheless, the couple specific surface area with this increase in surface energy, and you can start to understand why we saw what we saw previously, that particularly in the case of enteric dispersions, these particles are promoting, the spray dried particles are well, let me say it in the reverse way, because I like that better. KinetiSol particles promote congruent drug polymer release. They prevent drug release prior to the dissolution of the polymer. And I think when I was in graduate school studying amorphous solid dispersions, the idea was that you take an insoluble drug and you impregnate it with a polymer to where the drug's solubility and dissolution characteristics no longer look like the drug, they mirror that of the polymer. So by KinetiSol, we're holding true to that fundamental property of ASDs, where we want the drug's release to mirror that of the polymer. And with spray drying, you can see these differences that are driven by surface area and surface energy. So to carry the galeterone, just as a last case study, this is the graphical abstract from the paper. So I like it because in one slide, it just summarizes the whole thing. So we took galeterone, we made a KinetiSol dispersion in HPMCAS, we made the equivalent spray dried dispersion. You've seen these graphs. This equates to, once again, in vitro release, where the KinetiSol material prevents acid phase release and promotes intestinal phase free drug. And once again, the punchline, as always, we do an oral bioavailability study and shows superiority of the KinetiSol material relative to the spray dried dispersion. Because this is a propaganda presentation, as I said at the beginning, right? That's the disclaimer. Is this always the case? No, I think it's a majority of the case. It's particularly important when you've got drugs with rapid crystallization or precipitation kinetics, where the protective effect of the polymer is critical. If you've got a drug that can spring into solution and stay there on its own, they're rare, but they exist. We've seen a few of them recently. I think this is less apparent, and spray drying works as well as anything else. But for a majority of compounds that we see, the crystallization kinetics, precipitation kinetics are quite rapid, and polymer protection of the amorphous payload is key. And so I think this is a real effect for most compounds, that KinetiSol would outperform spray drying. So just a brief snapshot. I thought, it's nice to see sort of a graphical summary of where KinetiSol went head-to-head against solvent processing and showed bioavailability enhancement. So these are public examples that we can share. There are others, but I without going into detail, I think there are multiple examples that we've put out in the public domain. With the BI compounds, the Lilly, galeterone, our work with Zelboraf was particularly satisfying for me, coming from Roche. But to show that time and again, we show significant performance enhancement over spray drying, and it's rare that we're inferior. And if we are inferior, oftentimes it comes down to modifying the particle properties side, producing finer particles through modifying our milling process. Okay, so I think I've beat you all over the head with all of these points, so I don't need to summarize, but basically it's just saying that molecularly KinetiSol material and spray dry dispersions tend to have the same properties, but the particles are quite different. And I would argue for in the most cases, a lower specific surface area and surface energy promotes better performance. And there's the line of honesty written there. For compounds with rapid precipitation kinetics, KSDs tend to outperform SDDs. My final segment in why KinetiSol is the greatest thing since sliced bread is to compare KinetiSol to spray drying in terms of downstream processing, specifically tablet formation, because I don't think it's trivial that KinetiSol dramatically reduces the process flow and going from amorphous dispersion to final tablet. So spray drying is a series of typically 7 steps, starting with spray drying, secondary drying, addition of external excipients, which is a dilution step that I just sort of picked a midpoint. I think 25% external excipients at the roller compaction phase might be a nice midpoint. Could be less, could be more. Then coming out of roller compaction, you've got a sizing step, followed by additional dilution, adding external phase excipients headed into tablet compression. So I think from what we've seen with spray dry comparators that we've gone head to head with, tablets, as of midpoint, contain about 50% of the spray dry dispersion. Whereas with KinetiSol, we've eliminated 3 of these steps. There's no need for secondary drying, blending, and roller compaction prior to tableting. We come out of KinetiSol, go into a hammer milling step, a milling sizing step, and then adding the external phase excipients. Typically, 25% is all that's necessary to achieve a tablet with acceptable tensile strength and disintegration properties. So the final tablet, in many cases with KinetiSol, has about 75% ASD load, which can be, as I pointed out in some of the first examples, really critical in terms of reducing pill burden. And how are we able to achieve tablets with 75% ASD load? And in this case, the particles aren't blue. They came out of the mill orange, but that's not why they're compressible. The reason they're compressible, if you look at it, it's granules from a milling process. So under SEM, it doesn't look that much different than melt extruded granules coming out of a milling process. But if you look closely, there are pores in these granules throughout. And these pores are generated during the quenching operation where there's air entrapment. As you'll see tomorrow, when the material exits the compounder, it's got the consistency of Play-Doh, and then it's rapidly quenched through a compression operation to bring it down to close to room temperature. And in that quenching operation, some of the air is retained. And those pores are points for plastic deformation and brittle fracture. So that is what allows KinetiSol material to be significantly more compressible than similar thermally processed materials. So we have good compressibility with the KinetiSol granules, even without a lot of external phase excipients. So this is what allows us to, with minimal dilution, arrive at really acceptable tablets. Some examples of this are KDFX tablet. This is one of our most advanced products, phase 3 ready asset. It's a high melter, but we were able to create a 50% drug load dispersion by utilizing a combination of polymers, Orgegadel 155 and copovidone, that have different functions within the ASD, both critical to manufacturability and performance. And then Magsterate, which also serves as a processing aid, but we serendipitously discovered it's a bioavailability enhancement bioavailability enhancing excipient in this formulation. So we were able to get 50% drug load in the ASD, and then this ASD produced a viable tablet with what do we have, 80% of the ASD in the final tablet. So we have a 360 milligram dose in a tablet that weighs 900 milligram total weight. So that's a pretty significant payload for an ASD product. And it was absolutely critical for KDFX, the deferrosirox, because it's indicated for iron overload, and the doses go as high as 40 mg per kg. So pill burden is a significant issue for this therapy. Anything less than this dose wouldn't have been a viable product. So when the demand is for a high dose drug to reduce pill burden for an indication like this, we can meet the demand through KinetiSol. And this is just a table showing that we had good tensile strength and good disintegration. Similarly, we've done a lot of work with ritonavir internally and did some collaboration with AbbVie as well. And the driving force here is that, most of you know, ritonavir isn't used as a therapeutic agent itself. It's used as a PK booster in a lot of cocktails for HIV and other viral treatments. So the commercial Norvir tablet is melt extruded. I think the drug load is about 15%. I think it's about a gram tablet for the 100 milligram dose. So we aim to do the same here, increase the drug load in the amorphous intermediate, then increase the amorphous dispersion load in the tablet so that you can make something more amenable to fixed dose combinations or co-administration of ritonavir with other antivirals. So we were able to generate a 33% drug load, KinetiSol intermediate with equivalent performance to Norvir by pivoting polymers. So the commercial product is in copovidone, which isn't a great parachute for ritonavir. We were able to pivot over to HPMCAS, and what enabled that, ritonavir and HPMCAS are incompatible. Ritonavir is sensitive to acid catalyzed hydrolysis, but the cycle times of KinetiSol being so short, it prevents degradation due to HPMCAS exposure just by virtue of the short processing time. And then we were able to incorporate 75% of that dispersion in the tablet. So we fit a 100 milligram dose into a tablet that weighs less than 450 milligram. So this was a good example of how a case where a compound used in combination with several other products, pill burden's an issue, and we were able to pull two different levers in terms of drug load in the ASD and drug load and ASD load in the tablet to arrive at reduced pill burden. Oh, you can't see this at all. Oh, well. My buddy, where's my good friend, and he's not in the room, Jim D'Annunzio, he presented this graph that you can't see back in Denver in 2017, and he pointed out the issue with amorphous dispersions in terms of tableting, and that the comfort zone in industry for drug load was, he drew a line at about 40%, and then the comfort zone in terms of tableting for an ASD was about 60% ASD in a final tablet. So this was sort of the boundary layers for ASD products that were limiting the dose that you could achieve per tablet. And so the part of this that you can't see is with KinetiSol and our ability to utilize a combination of polymers and other excipients, we can often achieve higher drug loads with similar performance. So I kind of, based on deferrosirox and some other examples, like I put our boundary layer at about 50%. And then based on our number of case studies that show we can get acceptable tablets as high as 85% ASD load in the tablet. So the KinetiSol enabled zone for a typical tablet extends out beyond the typical where the industry is the industry's comfort zone based on melt extrusion and spray drying. So this is my last slide. So I think hopefully by now I've satisfied myself that KinetiSol is a simpler, more effective manufacturing technology than spray drying. Can I say it again? It's a much smaller physical and ecological footprint. There's two case studies where we showed where we think we're directly substitutable for commercial spray drying and can result in a bioequivalent product. And then for NCEs, we have the potential to provide much better performance than spray drying when we, just by virtue of different particle properties, but also by opening up a formulation toolbox that the other technologies don't have access to, we can arrive at an optimized ASD composition. And then the easy part, simpler downstream processing, that's obvious. And then we're able to achieve higher ASD load per tablet. And this is the breakdown of the assets we're working on currently. And as I said, about 3/4 of what we're working on is KinetiSol, is conversions from SDD. So when I said in the beginning that KinetiSol will disrupt spray drying, I think this is the evidence. The industry is looking for a better alternative to all of these problems, to deal with all the problems I mentioned, and it appears that KinetiSol is well positioned to be that alternative. And this is just the beginning. In the future, I suspect we'll put a significant dent in the spray drying market. Dave talked about stats, so here's my stats. And a lot of us in this room, we've been publishing papers on KinetiSol since 2009. Jim, who left the room, again, this probably makes him nauseous to hear it again. He published the first paper in 2009. Stephen's Galetterone paper was the latest in 2023. So we're beyond 30 peer-reviewed articles on the technology, and we're still learning about the technology. There's still tricks that we're working on and things that we don't know. And actually, now that the business model is such that we're trying to put the machines in the hands of other scientists, I think we stand to learn a lot more as we deploy the technology and put it in labs and let some of you big pharma scientists play around with the tool. And then so finally, just I get to present all this KinetiSol stuff all the time, and and I'm the face of it. But honestly, it's contributions from my team at AustinPx, and we have an amazing scientific team and an amazing leadership team and amazing financial backers, and it's a proud moment to stand up here in front of you all and present what we've been working on over the last 12 years and in a venue like this. I can't imagine you have any questions after that, but if you do, I'll answer them.
Thanks, Dave. Yeah, a quick question. What's the sensitivity of KinetiSol, generally speaking, to API particle size?
I would say in general, it's kind of insensitive to particle size. I think we designed the process parameters are developed according to the state of the drug substance as it's provided to us. I think the particle size distribution, the morphology is something that we track through development. Should there be any significant changes in particle size distribution or morphology, there'll be some tuning of the process required. That typically doesn't happen though, unless we start real early, like getting compound just fresh out of discovery. There will be some changes along the way. But by the time you get into clinical, it's been our experience that the drug substance properties are fairly consistent and the process doesn't change. But we've got the levers to tune should that happen. But it's generally insensitive. There's no upfront filtering of particle size or anything of that. Like we take the drug substance as it's supplied. Yeah.
I was just wondering, do you have a recommended drug substance quantity that is requested for the initial screen or the screening process using KinetiSol?
Recommended? I'd recommend a kilo up front. Do whatever we want. Well, so we've been making a lot of strides. Well, so first of all, I want to say that when we started out, all we had was the manufacturing scale equipment. So the requirements for API were more in the hundreds of grams, and then Chris designed and built the formulator, and that changed the game for us completely. And since then, we've been taking that formulator and working on some different methodologies to further reduce the amount of API that we need to establish feasibility for KinetiSol, and then allow us to do some polymer screens. So my team and led by Danny Davis developed a low drug load screening process where we can establish feasibility with the process in general, and then screen all the polymer chemistries with about 2 grams of
Awesome.
API. And we just finished a project recently with a ProTac compound where all they had was 3 grams, and we were able to do that screening and deliver a prototype ASD with just 3 grams. And
that is great information because it's a constant struggle in terms of what phase are you in, how much material do you have to spare. And I know on the analytical side, one of the reasons why we pushed the X-ray diffraction method of testing was because it was indestructible.
Yeah.
So again,
right,
same thing.
Testing. Yeah. Well, for us, it was you want to capture all the opportunities, and I think that upper left quadrant on that diagram, those brick dust compounds, we think when you're in that situation coming out of discovery where you've got a molecule that's ultra high melt and insoluble in everything, at an early stage, you're having trouble solubilizing the molecule to even get sort of readouts in a petri dish or early preclinical models. So there's a need for KinetiSol at a very early stage. So we wanted to try to capture those opportunities. When companies would come to us and say, the world supply of this API is 2 grams, what can you do? For a while, we were saying, well, come back when you've got 50 grams. But we've made strides to capture those opportunities, and we've been successful at doing that recently.
Great presentation. I really liked your slide with the 4 quadrants. It seems like it could be a solution for a wide range of compounds.
Yep.
I was wondering if there are any physicochemical properties or properties of a compound where you look at it and say maybe this wouldn't work.
Yeah, that's a common question we get asked. And The answer is no. I think in the early days, in the very early days when we were focused on enablement, we would look for high melt point, poor organic solvent solubility, as those were the opportunities that we wanted to utilize our bandwidth on, because it's not a situation where we could spend a lot of time developing a KinetiSol formulation and a company could just take that formulation and go make it by a different technology. So initially we kind of screened opportunities that way. But now, as we see the advantages with respect to spray drying that I've laid out here, and to some extent melt extrusion, there's really nothing up front that, thermal degradation, thermal instability of the amorphous form in the presence of certain polymers is something that we need to understand. But there's not a good surrogate for that. I mean, even doing thermal analysis, DSC, with physical mixtures, heat stressing, rotovap samples, for example, don't really simulate KinetiSol because the time scales are so much different. We're talking about 5 to 10 seconds. So this is why the low drug load approach has been so helpful, is that we can establish, like I said, compatibility of the drug with the process in general, and then its compatibility with all types of polymer chemistries really quickly as like a pre-formulation step prior to getting into consuming lots of API and heading down the development path. We just conduct this as a pre-formulation step, and we've had to in some cases, especially in early discovery where the APIs are less robust, you run this low drug load feasibility screen and you find out the drug cannot tolerate any exposure to heat in its amorphous form, and then we can quickly eliminate those compounds from consideration. So the answer is no, but we have a tool now that gets us to the data we need to screen out a candidate quickly.
Great presentation.
Thank you.
So like you mentioned, a lot of the drugs that you're using are rapid precipitators. So is there an influence on the processing of spray drying versus KinetiSol on the precipitation kinetics that you're seeing?
Yeah, I think that's what we've shown with a lot of with the in vitro studies of KinetiSol versus spray drying, is that it's the key is congruent drug polymer release to delay precipitation, delay nucleation and crystal growth by maintaining the amorphous drug in close proximity to the polymer throughout release. And I think that's where you see the impact. And also, it comes down to polymer selection as well too. The drug will have different affinities for different polymers in an aqueous environment, and in some cases, combinations of materials help in term. So there's also a formulation component too, where you're constrained to some extent by common organic solvent solubility. The polymer, the excipient system has to be commonly soluble with the drug. For spray drying, we don't have that constraint. We can mix anything into a single phase system using KinetiSol typically. And that's been effective for really difficult molecules that are really poorly soluble and precipitate really quickly. The ability to formulate creatively to find that synergy of excipients has been very helpful in some stubborn cases. But in general, particle morphology tends to help as a first step.